Hello, hello, hello. Am I audible to all? Hello. Am I audible to all? Yeah.
Audible.
Thank you. Thank you all. And good evening to all of my participants. And definitely good day and good morning to my US friends and, and all of my participants. Welcome to today international webinar on Down syndrome research, approach, and updates. This is organized by Trisomy 21 Research Society, Indian chapter, in collaboration with International Trisomy Research Society, Netherlands, and Department of Zoology, Cytogenetics, and Genomics Research Unit, University of Trisomy 21 Research Society, or is the first international non-profit scientific organization of the researcher across the globe studying Down syndrome. T21RS was founded to promote research on Down syndrome, stimulate translational research, and apply new scientific knowledge to improve the quality of life of the children with Down syndrome. The Indian chapter of Trisomy 21 Research Society is the first local chapter established in February 2020. It is inaugurated in the presence of Professor Stephanie Sherman,
error bars cross this point of 1. So what we see as risk factors, again, age by far is the most important risk factor. So increasing age is an increasing risk factor. This age was, here just notes about the adjustment for those who are status in her family survey. And then the other risk factors, we saw obesity, same as in the general population, and dementia or Alzheimer's, same in the general population. These are adjusted by the living situation, the level of intellectual disability, and by age, sex, data source, again, the survey, and the country of residence. So we think these are robust findings and have been adjusted for the appropriate covariates. Potential risk factors for which we need more data. Again, the increased risk among males, among those with seizures or epilepsy, though we need to understand this better. We don't know the age of onset. That's the next phase of the data to understand, are these early onset seizures or seizures essentially associated with dementia and the later onset? And then gastroesophageal reflux is a potential risk factor. We don't have evidence for those living in, if they're living in a residential care facility after adjusting for these covariates, or for the level of intellectual disability. So these are not specific risk factors. Similarly, having thyroid disease, obstructive sleep apnea, congenital heart defect, or behavioral psychiatric conditions are not risk factors at this point. We have no evidence at this point. We did start to look at the impact of vaccination on mortality. This is not the vaccination with respect to getting the infection, but instead on the course of the disorder, specifically fatality. And we see no indication that the flu or the pneumococcal vaccine prevent this severe outcome. But again, associated, these associations are adjusted for these specific covariates. But we want to say that this is a new part of the survey that we're delving into, and we'll present more data as we get larger sample sizes. So just because this is very important in terms of these pre-existing conditions, let me summarize this. The statistically significant risk factors are age, and as I said, older than 40, which is different from the general population, which we usually think of as older than 60 as a risk. Obesity, similar to the general population, and dementia and Alzheimer's disease, again, which is important because this is a very significant coexisting risk factor among those with Down syndrome. To see where these go once we get more data, being male, having the gastroesophageal reflux and seizures, we'll delve into those more as the data progress. And then, at least at this point, we don't see evidence for these being risk factors. So the take-home messages: monitor for the same symptoms as in the general population, potentially for the younger ones. Look out for nasal signs or maybe some of the enteric symptoms, but look for the same things as we see in the general population. We suggest that protecting those over age 40, so 20 years younger than what we see for the risk in the general population, and those, again, with the underlying conditions that I'll repeat here. So mortality is a higher risk for those in the general population and those for Down syndrome with respect, the risk factors being now age, obesity, and dementia, and we need more data to confirm the other potential risk factors. So please stay informed with that. We don't see any indication, though we'll keep looking at this as the data grow, for these listed here. And for brevity, I won't repeat them. The study limitations, I brought this up in the beginning. The first thing is that I didn't mention, we do have a large sample size. This is the good, the strength of this study is almost 600 cases now. But when you start to break them up into these categories, these questions that we're asking, the numbers are still small. So we need to really push to continue the survey to be completed over the next month so we can better understand how COVID-19, how the course of illness of COVID-19 plays out in those with Down syndrome. We're not able to make statements about the risk for this infection. We're trying to get more data from the general population, but this will be, the survey is really not set up to ask about the rate of infection, but we'll have to continue to focus on how to answer this question. And I think I mentioned this earlier, but we don't know whether those with Down syndrome are sent to the hospital at the same rate or for the same reasons. So we'll try to get more data to monitor this. And lastly, as I did mention, we've got to be careful because at this point, this is not a random sample. This is not a representative sample of those infected with the virus. So we have to make sure that everything we interpret is within that context. So I want to thank especially all the caregivers of individuals with Down syndrome. You know, this is a very difficult time. during the pandemic. So I want to just say thank you for all the things you're doing, but also for taking the time to filling out this important survey. Also, we want to acknowledge the whole task force. There's been great effort to really move as quickly as possible to bridge this gap of knowledge that we have and really move ahead to understand COVID-19. And importantly, we have a stakeholders committee that gives us advice and helps us with communication of the information that we learn. And so I want to thank them for their effort. If you want more information, there's a section on the website for the T21RS Society, which is T21RS.com, and you can see the section on COVID-19. And they'll keep up with the literature review as well as provide information about the results from the survey as time goes on. I also want to acknowledge, though it's just too long to list all these great foundations that have helped us with the implementation of the support for the implementation of this survey and for the data analysis. So thank you. I can take questions if there's time or whatever the organizers would like.
Thank you, Stephanie, for this excellent that you actually you highlight a lot of important things. I think participants, our students, faculty members, researchers, and particularly the caregivers and parents will get benefit from this information for taking care of Down syndrome children under these critical pandemic situations. Now it's a question answer time. May I ask Elena to open his camera? Okay, Elena, please open the question answer and chat box, and you please select some of the question to ask Stephanie, and Stephanie will be happy to answer a few questions from this. Stephanie.
Unmute. Wonderful. Thank you. I see that some of the questions have been answered as we go along, but there are a lot of important questions remaining. Let me start with one question that I had from the epidemiology here. I understand the limitations of this kind of survey in that you can't really measure what is the death rate or the infection rate or the hospitalization rate. Mm-hmm. But if any of those indicators were way off the scale, were very, very high compared to the non-DS population, you probably would have gotten some sense of that from your clinician survey. Did you?
Well, you're absolutely right. I mean, because even anecdotally, in the beginning, the task force had representatives from Europe and around the area. They weren't, it wasn't the huge crush we were expecting given the pre-existing conditions. And so that was the good news, not that different generation. I'm cautious with that, but actually right what we were worried about, the real, I don't want to say disaster, that's not the right word, but the a huge impact of the trisomy 21 with respect to COVID.
Thank you. Regulation in individuals with Down syndrome and how that might or might not play a role in the COVID response. Do you have any thoughts on that or any data that might?
So there was a very good webinar that T21RS just put on, did a beautiful job in summarizing the immune problems that exist among those with Down syndrome. So I mean, there's genes on chromosome 21 that are involved in the immune system itself. Like there's 3 or 4 interferon genes. There's microRNAs that regulate other genes involved in the immune system. So it turns out that there's, like at the cellular level, at the systems level, at both those levels, and in the innate immunity and the adaptive immunity, there are dysfunctions going on because of that dysregulation, primarily, I think, because of the dysregulation on trisomy 21 of those genes. So there's how those play out, and Mara might be able to answer this better because she's done some transcriptomics, but how those play out with respect specifically to this virus and its illness. There are some good data that are helping us understand that. I'm not an immunologist, so that was a bit of a waffle there. But Mara, if you want to step in and say something specific about that, please do.
Yeah, so as Stephanie was mentioning, we did a meta-analysis, and maybe I can share my screen so that you can kind of see this. Let me just, can you see the whole screen? Mm-hmm. So we found some molecular processes of susceptibility. So like, the overexpression, the upregulation of TMPRSS2 that is priming the spike protein and favoring the entrance of the virus, into the bad. And of course, the hypothesis is that the increased would, of course, drive a more severe cytokine storm. And there are other susceptibility factors. But on the other hand, there are also kind of molecular processes that might be protective. And this might be part of the answer to a previous question of why don't we see so many cases. As Stephanie said, we have to be very cautious because we still don't have sufficient cases to really make a statement. But it's true that given all these inflammatory response and other factors that are maybe less known that might help increase susceptibility, reduce the impact of viral genome release, and also the resistance to the apoptotic effect, of the virus, right, through the AKT signaling. And then we also have some factors that might help reduce viral replication. So as Stephanie was mentioning, there are many factors, and we still don't understand. I mean, the transcriptomic data are suggesting some possible pathways, but we are still working on trying to understand how they are confluencing to really drive the final outcome of the infection.
Thanks, Mara.
Thank you. Professor Ghosh, should we continue with questions or move on to the next speaker? You're muted.
You are muted.
Yeah. Sorry. Eleanor, please ask 1 and 2 more questions from the chat box or question answer box. So you may ask.
Wonderful. Okay. There are also several questions about blood group as a risk factor and whether that is important or something you look.
So we didn't, unfortunately, when we started this. We didn't want to put lab outcomes as well as blood group, but that seems to be becoming an association in the general population, as I understand it. So, but we don't have those data. Sorry.
Let's see. And also questions about ACE2 receptors and genes in chromosome 21, whether there are any differences, whether there are any differences in chromosome 21 genes.
No. No.
And also a couple of different questions about the sex difference and whether there's
why males. I don't know the difference. Again, why more males are affected. I don't know if there are more underlying comorbidities among males that are not being picked up, or what the answer is. Again, I could. Mara, have you heard a good explanation for why males are?
Not a very good one, but it's true that in Down syndrome is also upregulated. For example, in some male-specific disorders like prostate cancer and so on, and it's regulated through testosterone. So it has been suggested that this might be one of the factors contributing to this increased male prevalence, right, and severity.
Okay.
Shall we take more questions?
Yeah, one question from YouTube live chatting. May I ask this question to Stephanie? This question is there any correlation between Down syndrome and any kind of viral infection? I think almost related to immunity and everything.
Yeah, I mean, so just the idea that there is an increased risk of respiratory tract infections. But that's back to, let me just see if I can refresh my memory. Yeah, so I think there also is an increased risk for respiratory syncytial virus, the RSV. I'm looking at the data that was done with the literature review, and H1N1. So they have a higher risk for those viral infections specifically, but again, the other infections, at least with the respiratory tract, could be more the bacterial.
Excuse me, there is a good question from Shaunak, I think, in the question answer section. Have you taken this question? Probably not.
Yeah. Eleanor, can you please search the question from Shaunak Shahu from NIH? It is in the question answer box. May I ask this question on behalf of you?
Please go ahead. I can't find it.
Shaunak Shahu from NIH is asking, are there any new mutations in SARS-CoV-2 genome identified in DS children to virus, and any association with ACE2 receptors and the gene on chromosome 21?
So that's, which makes them more susceptible. Again, and then the second question, any association with ACE2 receptors and genes on chromosome 21? Again, I think, not that I know of. Again, Mara, can I pass that to you?
Sure. I mean, we haven't seen overexpression of ACE2 except in iPSCs in Down syndrome when we look at the meta-analysis of all the transcriptomic data. So the ACE2 receptor is not differentially expressed in Down syndrome samples. Mm-hmm. But of course, the fact that some priming proteins like TMPRSS2 is there overexpressed might be, and in fact, TMPRSS2 has been proposed as a possible target for therapies, because if you have it upregulated, you may increase the priming of the spike protein, and this influences the entrance. And in fact, cathepsin B, is not in Down syndrome. So TMPRSS2 SS2 should be the main way of entrance, right? So I think more than ACE2, it's TMPRSS2 in Down syndrome. But again, this could mean that therapies targeted to TMPRSS2 might be more useful for people with Down syndrome, right?
Mm-hmm.
Very interesting.
Yeah.
I think okay. Actually, it's a group work that we are doing.
Definitely.
It's a task force.
It's a task force.
Okay. Now there are so many questions, and may I request participants to stay online. And I think it will be better to answer all those questions. May I request Professor Sherman to discuss regarding this question in person. And after getting your feedback, I could put all those answers to the participants, because now we are going out of time. It's a slot for Mara Diersen, the next talk. So Stephanie, once again, thank you very much for your excellent talk, and so much informative. That was really, thank you. And on behalf of the entire team of Trisomy 21 Research Society Indian chapter, I would like to congratulate you and express my greatness to you. Thank you.
Thank you so much. Thank you.
Okay. Now it's Elena. Please do introduce Professor Mara Diersen.
Wonderful. We have already heard some very informative comments from Dr. Sir. Dr. Mara Diersen Sotos she's presently the group leader of cellular and systems neurobiology in Tabria in 1990, and her postdoctoral work at the prestigious Optimistas Comprometidos Prize in 2018 and the Big Vang Medal in 2017. She has been elected as president of the Trisomy Research Society for the 2016 to 2019. Her lab focuses on understanding cognition and behavior and how they work in cases of intellectual disability like Down syndrome. She's interested in what are the changes in neuronal architecture and connectivity that disrupt cortical and hippocampal function in these genetic cognitive disorders. In other words, what's the multi-scale link from molecular alterations to impaired cognition and behavior? And I'm looking forward to hearing more from her today.
Thank you very much. And well, I will share first my screen to see if it works again. Let me just. Why don't we see so many? As Stephanie might be one of the factors contributing to this increase. I hear speed and severity. Okay, so can you hear the screen, or I think it's Dr. Ghosh, maybe? Okay, good. So I muted him. Sorry about that. So first of all, thank you for inviting me to this webinar. And I'd like to say that, especially Sujai, thank you very much, and the whole Indian chapter of the Trisomy 21 Research Society. I'd like to thank you all for your strong commitment to improve and help advancing research in Down syndrome. And I have to say that your chapter is one of the most successful chapters, to not say the most successful chapter in Trisomy 21 Research Society. So it's a
thank you, Mara. Thank you for your acknowledgement.
No, it's absolutely true. Nothing to thank, but it's really an honor to be here. So what I decided to talk about was about our work on trying to unravel the mechanisms of intellectual disability in Down syndrome. And yeah, let me see if I can move. Okay, good, I found out. So my lab, as Elinor said, thank you, Elinor, for the introduction, is located here in this absolutely beautiful place, just near on the beach, right? And we are the Cellular and Systems Neurobiology Lab of the Center for Genomic Regulation, which is a biomedical research institute. And those are the people from my lab. As you see, the gender balance is not very good, but I hope it will improve in the next years. So we are interested, as Elinor also mentioned, in understanding the mechanisms of learning and memory and how are those altered in intellectual disability, and specifically in Down syndrome. We have mostly contributed to Down syndrome research. And this is a challenge. Why is it a challenge? Because we are dealing with a disorder in which we have 3 copies of a whole chromosome. This means that we have a genome-wide gene dosage disequilibrium, right? And this is driven by the trisomy, but also affects other genes in other chromosomes. And this is something that some people from my lab, Hilario Thomas, Cesar Sierra, and Miguel Sabariego, are trying also to investigate. So we have observed this deregulation, this genome-wide deregulation in many different tissues and in many different situations. And Hilario did a whole meta-analysis of all the transcriptomic data that are available. So what happens? Okay, of course, the problem is also the second challenge is also that the neuropathology in Down syndrome is multi-scale. So this trisomy drives molecular alterations, but those have an impact on the cellular level, on the circuit level, and then on the whole brain function, right? And in fact, when we look at the phenotypes in humans with Down syndrome, what we see is that we have a macroscopic phenotype that is mainly characterized by a microcephaly, but this microcephaly is specifically or more directly affecting some structures like the temporal lobe or the cerebellum and also the cerebral cortex. So here you see some images of the hippocampus and how it's reduced, and specifically reduced in volume in Down syndrome. Not only that, when we go further on and we look at the microscopic level, we see that the neurons, and specifically the pyramidal neurons I'm mentioning here, but it also affects other neurons like interneurons, right? But what we see in the pyramidal neurons, which is the most important excitatory neuronal type in the brain, specifically in the cortex and the hippocampus, which are regions involved in learning and memory, what we see is a reduced complexity and also a reduced size of the dendritic tree. And also, we see a reduced number of dendritic spines that are the points of connection, so the points where the information is trespassing from one neuron to the other in the brain. And in fact, all these phenotypes come from molecular alterations in different pathways that include many different functions like synaptic vesicle endocytosis, vesicle-mediated transport, seal adhesion, neuronal differentiation, etc. Okay? So, in my lab, we use as an experimental tool the mouse models of Down syndrome. And mainly, we have been concentrating on a mouse model that bears a trisomy of part of the chromosome that is orthologous to chromosome 21 in humans, which is chromosome 16 in mice, right? And in the trisomic mice, in the T65DN, we have a third copy of the chromosome that is composed by most of what we call the Down syndrome critical region, which is the minimal region that when in triplicate gives rise to the phenotypes of Down syndrome. Interestingly enough, these mice also have some genes that do not pertain to the orthologous to chromosome 21. So to say so, there is not all the genes that should be there, and there are some genes that should not be there in triplicate. However, when Hilario and Cesar and Miguel looked at the data, all of them found that what is mainly differentially expressed are those genes in chromosome 21. So validating the model, right? And also interestingly enough, mouse models of Down syndrome recapitulate the Down syndrome phenotype. And what we see is learning impairments, and this is a task in which mice have to escape from a pool in which you have a hidden platform, and this is called the Morris water maze, okay? So wild type mice very fast learn how to escape, right? But trisomics have more problems. So they have really a learning impairment. And also in other types of memories, such as the recognition memory. So they have to recognize if an object of two objects that are presented is new or not. And this is, we measure this by the discrimination index. This is a very important memory type, right? So basically what we see is that this is hugely altered in trisomic mice. This comes along with alterations in the brain size, recapitulating this microcephalic phenotype in humans, but also with specific alterations in the dendritic tree morphology, right? So the neuronal architecture is significantly altered with reduced size and complexity of these dendritic trees. And interestingly enough, these will lead, we hypothesize, to a wiring perturbation. So the most important thing in the brain is how neurons are connected to each other. And they connect through these elements, right? So the dendritic trees are the communication structures of the neuron. And what we have seen is that this doesn't happen only in Down syndrome. It happens also in other mental disorders or intellectual disability disorders, and also in neurodegenerative disorders. So it happens in Down syndrome, in Fragile X, in autistic disorders, Rett syndrome, schizophrenia, and also Alzheimer's disease. In all of them, we see an alteration in the general shape of the dendritic trees and the dendritic spines, right? So basically, let me just put my clock so that I don't go too long. Basically, what we thought is, okay, why these should alter cognition, right? Why? I mean, you could have smaller neurons, but function the same, right? So what we know is that cognition and behavior are emergent properties of underlying neural circuits, right? So basically, the elements that are kind of driving or defining how the system works are the neural architecture, because these elements are the communication elements of the neuron, right? And these will modify neural physiology. So how a neuron is connected will define how this neuron works. And these will define also how the circuits, these overlapping circuits that are underlying cognition and behavior are working. So it will depend on how the neuronal structure is, you will have different functionalities. And this is fine-tuned by neural plasticity, okay? So through plasticity, you may change the shape of these dendritic trees and modify how the system works. So the idea is that if we have smaller neurons and less connected neurons, in the end, this will reduce also the capability of the neuron to perform a normal operation, right? But of course, this was a hypothesis. So we had to demonstrate that. And Linus Manuvens in my lab, Linus is now PI already in China, in Nanjing, but he was studying to what extent Down syndrome neurons were suboptimal. So he concentrated in specific properties like the routing information, so how the information is routed towards the soma of the neuron, and also this, what we call the storage capacity, so the capability of the neuron of identifying different patterns, input patterns, right? This is very important. If it's a time-dependent pattern or a place-dependent pattern, etc. And then the cost, because of course, there is a trade-off between the cost of the dendritic tree and also the capacity of the dendritic branch. So he did a whole statistic with new metrics that have never been used. And he found that there is a strong alteration in different parameters in Down syndrome mouse models. So this is a trisomic and this is a transgenic, right? And why this is important? So, of course, how does single cell morphology influence the network? So we cannot ask this question directly, so we had to use mathematical modeling. And Linus constructed these models to explore the topology of the cortical network. And in this model, the nodes are the neurons, right? And the edges are the contacts between the neurons. So this is a weighted network, so more synapses are taken into account by increasing the edge size, right? And what he found doing a whole exploration of different dendritic architecture, what we call a Pareto front. So these would be the set of configurations that maximize dendritic tree functionalities at a given cost, right? And there we could map different properties of our neurons, like branch density, tree size, dendritic straightness, and we could also map onto those Pareto fronts, right, defined by those metrics, we could map where our neurons would be, right, if they were optimal or not. And in fact, what we saw is that whereas wild-type neurons were mapped onto the Pareto fronts, in fact, those defined by synaptic contact probability here in brown, and dendritic tree size in pink, right? And here we have also the dendritic tree size variability. So what we found is that, in fact, neither trisomic nor transgenic neurons were optimal, and the network was also not optimal, right? Routing efficiency and storage capacity in the network was reduced due to these single cell morphological aspects. And of course, this would have, we predicted that this would have an impact on the network functionality. So the neuronal network topology would influence the functionality. And we did some experiments, we did some experiments with a group of Mavis and Vives, and specifically with Marcel. And what we did was do these recordings, with these multi-array recordings in the cerebral cortex and the hippocampus, also with Maria Alemany in the Vicky Puig lab. And what we found was that, in fact, the cortical excitability measured by a relative firing rate was significantly reduced in transgenic and trisomic mice, but also the oscillatory patterns, and specifically the gamma frequencies, were significantly reduced in the transgenic mouse model. And this is very important because those oscillations reflect the fast synchronization between neurons and are related and are involved in cognitive patterns, right? So basically what we found was that these architectural features of Down syndrome neurons were driving a network functionality that was suboptimal also, and this had a specific consequence on firing rate, excitability, and oscillatory patterns. So we decided to study how these would influence behavior and specifically memory. But we wanted to study these in an yet unexplored, let's say, aspect, which is what we call engram pathology. And what is engram pathology? So let me go back to the definition of Richard Semon in the 1920s. And he defined these enduring physical and chemical changes, leading to lasting connections in the brain that result from simultaneous excitation promoted by learning. So this, in his hypothesis, would underlie memory formation. And in fact, translated into the neurobiological aspects of memory, what we could think of is that when you learn something here, like you see the Eiffel Tower, an ensemble of sparse neurons that get activated. And those neurons, because they are simultaneously activated, strength of their is not used when you are not remembering these specific issue, right, like the Eiffel Tower. But when you remember it, what you do is you reactivate exactly this specific ensemble of neurons. So these engram neurons, right? That are forming the engram and recovering some memory. So how he did that? So the idea is we have this basal level in which all the neurons are not activated. When you learn something, you activate some specific ensembles of neurons, right? That then Tonegawa was able to label molecularly, right? So this molecular labeling allowed also artificial reactivation. And what he discovered was that by reactivating these neurons that form the engram, he was able to recover the memory that was related to this specific engram, right? So this is great because these new engram tagging strategies have opened a whole new aspect of what is memory and how can we kind of influence the memory performance, right? So in my lab, we have used a strategy that was also used by Tonegawa, that is capitalizing in the fact that when neurons get activated, like here, right, they express immediate early genes. CFOS, ARC is more specific of memory, and they are upregulated, right? So that by profiting this fact, you can then tag these neurons. And Álvaro Fernández in my lab did those experiments. So basically, what we use is a chemogenetic strategy. What is this? This means that we are targeting these cells that express CFOS, right, upon learning. And what we are doing is we are tagging them with a marker like mCherry, but we are also inserting in their membranes what we call these designer receptors. So the designer receptor is exclusively activated by designer drugs or DREADs, are specific molecular entities that only respond to a designer drug. So you put them into the neurons, and then when you inject these drug, they will get specifically activated by that. Okay, so basically what we can do is we can tag the engram neurons, but we can also manipulate them. So this is a strategy that we use to define what could be the engram pathology in Down syndrome. And specifically, just let me recap, what we do is we tag the engram cells, but we, and those are activated by learning, but we also are inserting in their membranes these threads, so we can excite or inhibit the engram cells by injecting CNO to our mice. And thus we can either reactivate the memories or suppress the memories, okay? So we can play there a little bit. Okay, so first thing that we had to prove is that the fear memory was also impaired in trisomic mice. And how this works, so we put the mice in a context that is not dangerous, let's say, and of course they are very active because we measure the fear response by the freezing response. So when you get a very kind of strong fear stimuli, you freeze like this, right? So this is what we do in the context B. So what we do in this second context is the training. So we are providing shocks, mild shocks, but a long time. And what we see is that the wild-type mice, they learn very quickly, oh my goodness, a shock is coming, right? So they freeze more. And instead, the trisomics are worse and significantly worse in learning the task. But this is also reflected when we put back the mice in the context B, in the context that was trained paired with a shock, but this time we don't give a shock, and the mice, the trisomic mice, remember less that this was a fearful context, it's a dangerous context, right? So Alvaro found that, in fact, this was accompanied by a kind of quite strong increase in the number of cells activated. And here we measure these activated cells by the expression of C4s, right? And of course, in non-learning conditions, so in the context that is not dangerous, they have more or less the same activation of the neural cells. But when they are exposed to the fear conditioning context, then the wild-type mice, after learning, they show an increase, a significant increase in the CFOS positive cells, which is not so high in the trisomic mice. When we look at a more specific marker of learning, like ARC, here you see that the result is even stronger, right? So the activation is much less in trisomic mice. And this is important because if you dedicate too few cells in your engram for a specific memory, this risks the enduring integrity of memory, right? And this may be due to different things. You could think, okay, maybe it's simply because they have less neurons. Okay, we check this, and this is not the case. Alvaro checked this, and in fact, we don't see significant differences in the number of cells in trisomic mice. So this reduction of activity, good. And then we thought, okay, let's play a bit. Let's try to reactivate the memory. And to do so, what we did was an experiment in which you can see if you can reactivate the memory in a non-fearful context. So we habituate the mice, and here we have the non-fearful context, but then we train the mice, so we give the shocks in context B, and then we go back to context A. So in principle there, you should not have any memory, or if something, you should have a memory of the safe context, right? But there we can artificially reactivate the engram because we have these dreads expressed in the engram cells, and we can ask the question if activating the engram cells is sufficient to artificially reactivate fear memories. So we did so, and here I put you how they behave normally in context A, right? And now we will reactivate the memory. So the same mice, right? When you reactivate the memory, okay, look at that. The wild types have a almost complete reactivation of their fear memories. So we have created a false memory by using our engram activation, right? Instead, unfortunately, the trisomics also increase their freezing behavior, but much less. And this is very similar to what happens in normal conditions. So the wild types almost reach the same levels of freezing than in the natural recall tasks. So when we don't reactivate the engram here, but the trisomics also retain the same deficit that they had in the beginning. So even reactivating the engram doesn't allow to recover the memory deficit. And this suggests that it's not a problem of reactivation. It's a problem of engram allocation. So it's a problem of how many neurons do we have in the engram, okay? And in fact, when we look at the CFOS M-cherry cells, which are the real engram cells, before we just looked at the CFOS cells, but now we look at those cells that were tagged in the engram. And those are almost significantly reduced. We are still doing experiments, so this is fresh result yet. But yeah, what we think is that the reduced allocation to the engram results in these learning deficits, right? So what are the mechanisms? We are still doing the experiments, and unfortunately, I don't have much more time to explain you all these results, but you can ask for that if you want. So basically, we have some hypotheses, right? So we think that because it has been shown that the allocation to engrams depends on excitability, so you need to have excitable cells to really allocate them to an engram. We think that the reduced excitability that we see in trisomic mice, and this reduced excitability is contributed both by the increased expression of potassium channels that are overexpressed in Down syndrome because they are present in chromosome 21, and also the reduced activation of phosphocrep that we saw in previous studies could contribute to this reduced excitability and to this reduced engram allocation. But also the reduced LTP that has been observed by our group and by others, all of those could lead to a pathological or defective engram, and this could lead really to memory deficits. So one thing that we are also exploring is to what extent some specific genes might contribute to these phenotypes. And in fact, we showed previously that DIRC1A was contributing to some of these phenotypes and also to the dendritic architecture phenotype. That is also part of possibly the cause of this reduced engram allocation. So to what extent can we rewire these neurons, the Down syndrome neurons? So to look at that, in fact, what we have done is try to normalize the dosage of some candidate genes. I here will talk only about DIRC1A, but of course, we are also going to explore other candidates, and we have explored those in the past. But I talk about DIRC1A because we already have a drug that targets DIRC1A overexpression, right? And that might rewire the whole circuits, right? And this candidate is, as I told you, DIRC1A. So DIRC1A is a kinase that is phosphorylating many very interesting proteins, alpha-synuclein, dynamine-1, synaptoginin, cycling L2, tau, APP, and may contribute to some learning deficits in the mouse models. And in fact, when we overexpress DIRC1A in a transgenic mouse models, we already recapitulate the phenotypes that we see in the trisomic mice, like the learning deficits, but also the neuronal architecture deficits. And this means that DIRC1A is sufficient to produce the phenotypes when overexpressed alone, right? So we think that normalizing DIRC1A could be a very good tool for dendritic rewiring, because as Melchelle Pons in my lab, who is also not anymore in my lab, she's a postdoc in IRB, in an institute, very prestigious institute in Barcelona. But with her, we normalized genetically with short hairpins, and also pharmacologically with a drug called epigallocatechin gallate, that is a green tea catechin. We normalized DIRC1A expression and activity. And I will show you some of these results. I don't have time to go through everything, but what we found was that when we administer EGCG for 1 month to transgenic mice, the increase in the kinase activity of TL1A in the hippocampus is significantly reduced and normalized to wild type levels. And this is accompanied by a complete rescue of some of the memory phenotypes. So here you see again this discrimination index, so the memory recognition memory deficit that is very strong in transgenic mice overexpressing only DR1A, and that is fully recovered by the treatment with epigallocatechin gallate. Not only that, but when we look at the brains of these mice, in fact, what we see when we compare the wild type with the transgenic mice, we see these deficits that I was explaining to you, and this is stratum oriens in the hippocampus. So dendrites are shorter and less connected. And with epigallocatechin gallate, as you see, we recover completely these phenotypes, right? So normalizing DR1A expression or kinase activity improves cognition and also leads to a dendritic remodeling phenotype. And in fact, we went, thanks to the collaboration of Rafaela Torre, to perform several clinical trials with adult persons, or also a pediatric study that we are still conducting. And what we found was that individuals with Down syndrome that received epigallocatechin versus cognitive stimulation versus those that only received cognitive stimulation are much better in a global clinical improvement index that I can explain later on if you need. And interestingly enough, we wanted to have some kind of more biological insight into what was going on. And here you see some experiments with functional MRI in which we were looking at the activity in baseline conditions upon this task in which they have to sort out which is the piece that is missing. And what you see is that in baseline conditions, they mainly activate the visual cortex, right? After treatment, you see that besides having a higher activation, what we see is that they also recruit more cognitive related areas like the cortex, the frontal cortex, and also the hippocampus. And this is accompanied by an increase of functional connectivity, meaning that probably we are really increasing the plasticity in these brains, right? And we have some biomarkers that tell us that DR1A might be one of the candidates to explain this phenotype. But of course, there are many other things that EGCG is doing. And if we look at the disturbance of the proteomic network driven by DR1A overexpression that is altering many different cascades, what we see is that our treatment, and in fact many of those proteins are related to intellectual disability, what we see is that the treatment with EGCG rescues many of the most important hubs in the DR1A proteome, right? So in conclusion, what we think is that both procognitive treatments, both pharmacological and non-pharmacological, like environmental enrichment, are boosting environment or activity-dependent plasticity, and thus network rewiring. And what we think is that, of course, this change in the dendritic morphology is somehow leading to a, let's say, optimalization of the neuronal phenotype that would put these neurons in this Pareto front of optimality, and thus would contribute to rescue the network activity and the engram allocation. And with this, I'd like to finish first by saying that we think that contrary to what has been said during many, many years, understanding the neurobiology underlying cognitive alterations in Down syndrome will certainly lead to most efficacious pharmacological and non-pharmacological intervention. And we think that these multi-scale treatments are the answer to the cognitive disturbances in Down syndrome. But I also think that even very small changes in intellectual capabilities are really, really strong drivers of integration for the Down syndrome population. So with this, I'd like to finish by thanking again my lab, the funding sources, and also my good collaborators, Rafael Torre and his team, and also people like Jordi García Ojalvo that has contributed to the computational models, Eduardo Sabido to the proteomics, and James Sharp to the neuroimaging studies. And with this, I'd like to just finish with a sentence of the BRAIN initiative that we are funded with, that says, the time is right to inspire a new generation of neuroscientists to undertake the most groundbreaking approaches ever contemplated to understand how the brain works and how disease occurs. Thank you very much.
Eleanor, are you online? Eleanor. Eleanor. Okay, so it's a very, very nice presentation, Mara. Really, we learned a lot of new thing, how to decipher and how to approach a complicated subject like memory and cognitive ability. And there are a lot of questions. We received a lot of questions. And sure, I just like to ask Dr. Papia Ghosh to conduct the question answer session. Dr. Ghosh, would you please conduct the question answer session, taking question from the chat box and also from the question answer box? Dr. Ghosh?
Yeah, I can do it. Thank you, Dr. Ghosh. There are so many pertinent and relevant questions, Mara. First of all, there is a question from Dr. Shubhit Dashgupta that manipulating engram cells.
Yeah.
By manipulating engram cells, shall we manage depression in these particular cases?
In Down syndrome, he means. I don't know if there is many studies of how the engram changes, but with depression, right? But I'm sure that it will change because we see cognitive alterations in depressive patients, right? So I think that, in fact, it would make sense that it does, frankly speaking, because if you are modulating the engram during depression, as, for example, also in stress-related disorders, right? So post-traumatic stress, for example, could be an example. Using the engram strategy, you could inhibit the engram cells that are related to this deleterious memory, for example. So why not in depression? I think that also helping these ensembles to work better should help. But I don't think this is a nice idea, because I don't think there are many studies on that.
Thank you. Another question, Dr. Dashson, for you. How close this model that you are describing actually mimics human DS development?
Down syndrome development. Okay. So in fact, we are precisely...
This is a good question.
We are precisely studying this. I mean, as I said, there are several constraints, right, with mouse models. On one side, there might be species-dependent phenotypes differences, right? So what we are now comparing, and this is done by Miguel Sabariego and Cesar Sierra in my lab, and also with the help of Laura Batlle, we are comparing the development of organoids, brain organoids, and this is compared to embryonic development, right? So the thing is that we have to think that also for example, in the trisomic mice, we have some non-chromosome 21 genes that are overexpressed. Right? So this might also constrain the possibility of comparing. But even so, I think that from what we saw, we are recapitulating many of the phenotypes that are seen in Down syndrome. For example, there is more difficulty in recapitulating some neocortical features, because of course the neocortex in mice is different and develops in a different way, but still we have seen many parallels in the pathological development between Down syndrome fetus and Down syndrome mouse models.
Okay, you have explained very nicely. So many questions. Should we continue with one or more? I would like to ask you.
Yeah.
Okay, Professor Dyerson, then another question is, does epigenetics contribute to this disease development?
Mm-hmm. Well, that's again a great question. I'd love to have more time to go into everything. But yeah, so in fact, in the lab, we are also exploring this possibility. There are several genes, including DRP1A, that might modify the epigenetic control, right? So what we have seen, and this is the work, the thesis of Cesar Sierra, what we have seen is that there is a general hypoacetylation in Down syndrome. So basically what Cesar did was to try to see if he could recover this with a drug called SAHA. And in fact, when you use SAHA, you recover the acetylation. So meaning not only that, yes, there are epigenetic factors involved in Down syndrome, but also, yes, these epigenetic factors might be involved directly in the learning and memory phenotype. So the answer is yes, definitely.
Okay. One more question is, neurogenesis and neuronal apoptosis seem to be the major causes for reduced number of neurons. Mm-hmm. She is asking, is there any role of glial cells and inflammation in this particular issue?
I mean, thank you for all these questions. I mean, it looks like you have looked at my project. So in fact, we have among the attendants is Álvaro Fernández that is specifically looking at the astrocyte phenotype. And the idea, it's so interesting because, you know, we have this astrocytosis, this astrogliosis in Down syndrome. And in fact, you know, we have overexpression of S100 beta also, and we see this in this astrogliosis phenotype. So there is a gliogenic shift during development that drives this increase in the relative number of astrocytic cells, right? But also they are expressing more S100 beta. So of course, this could be interpreted as a mild astrogliosis, but what we asked was, okay, yeah, but how does this contribute to learning and memory phenotypes? And there is a very interesting new idea that the neural transmission is not just a neurocentric phenotype, but is also strongly contributed by these astrocytic cells, because they are modulating how neural transmission occurs. And the interesting thing there is that each astrocyte may control different neurons, right? Different synapses, let's say, right? So these somehow might pose the idea that these astrocyte neuron domains that are kind of common across several neurons, one possibility would be that they might help synchronizing the neuronal activity, right? Because they are really targeting several neurons. And this is very stable. So we see that we have one astrocyte contributing to control several synapses. Okay, so what will happen when we have an increased number of astrocytes, like in Down syndrome? How will this really affect neural transmission and learning and memory? And this is something that Alvaro is asking also by targeting specifically the astrocytes. So we have seen several phenotypes like reduced calcium oscillation in the Down syndrome astrocytes, etc. But what he's now doing and we will see how the experiment works. But what he's doing is, with the same engram tagging strategy, he's targeting the astrocytes that are activated during learning. And then we will see if by inhibiting the astrocytes, we will reduce the activation of the engram or not, or if by increasing the astrocyte activity, we can recover the Down syndrome phenotype memory, right? Memory phenotype, sorry.
Thank you. So many questions. I think later it can be addressed, because a lot of aspect, and your work is such a nice one and unique one in the field of Down syndrome especially. So a lot of questions are coming. Everybody is getting so much interest in particular this. So Dr. Ghosh, I think we should pursue with all these questions
one
later.
and two questions. We have to ask
you
one
can.
and two questions taken from the live chat of the YouTube as well. And Professor Shubhra Kumar Hui from the University of Calcutta is asking, is that possible to check dominant negative version of DYNK1A could rescue the dendritic spine phenotype in DS mouse?
In fact, we did this with short hairpins. And what we found was that we could rescue both the cognitive phenotype and also LTP phenotype, and in part also the dendritic phenotype. So what we have, of course, why did we go into EGCG that is more unspecific and so on and targets many other things? Simply because we needed something that was useful in patients. And this is why we moved from the gene therapy strategy to the pharmacological strategy. But yes, it did, in fact, it did rescue. This is not to say that DYNK1A is the only answer, right? So then what we think is that DYNK1A is like somehow a pathway disorder. And this has been shown by Hilario de Toma with his meta-analysis. So basically, it's not just the chromosome 21 genes, but also the chromosome 21 gene interactors who are contributing to those phenotypes. So probably what we will have by targeting only DYNK1A is a partial rescue of the whole phenotype. But certainly it's one of the best candidates we can think of. And there is a lot of labs developing more specific DYNK1A inhibitors, and there have been many, many papers, and companies also like Perhac in France is a company that is devoted to develop DYNK1A inhibitors, in fact.
Yeah. And last one, Mara, it's from my sides, actually. I am curious to know, actually, maybe it's very silly, because Down syndrome children, they exhibit their incapability in mathematical skill and calculative ability. But at the same time, they could memorize the music and some cultural work. They could perform the dance, could sing well, and they have a strong sense of music. Is it really related to any brain structure or special neuron?
So, sorry, I have muted somebody.
Yeah.
In fact, if you look at it, this phenotype is pretty similar to some autistic patients, right? They have they may have some increased or decreased mathematical abilities with increased or normal musical abilities. And this is very interesting because somehow what, let's say the hypothesis would be that the loss or the retaining of some connectivity patterns would be responsible for that. Like this has been demonstrated for some specific phenotypes, like synesthesia, for example, right? So you have this brain side for numbers and the brain side for colors that are there and that are connected when you are growing up during development, but then you lose this connection, right? So somehow there is a, let's say, structure-specific pattern of connectivity that is lost or not, or enhanced in these individuals with Down syndrome or with ASD, that might explain some of these features. And in fact, if you look at lesion phenotypes, there are some cases of lesions in which you get increased abilities in some very specific aspects, right? So I think it's still a lot to learn there. But I also think that by really making an effort and looking at the connectivity pattern, for example, in Down syndrome, compared with ASD or with other intellectual disability disorders, but specifically ASD because they have these savant syndrome cases also, right? So these might enlighten a little bit what could be the underlying phenotypes.
Thank you, Mara. Thank you. We are not dragging this session more. There are a lot of questions, a lot of curiosities, but I will deliver all the questions in person through mail. And after getting your answer, I will return back to my participants and try to satisfy them. Once again, we are thankful to you for your kind consent to attend this seminar as a speaker and manage some time to spend with us. Thank you. Thank you, Mara. We will meet again in the next meeting of our task force. Okay.
So I would like to also thank you. I'm really impressed by the number of participants and by your leadership, the Indian chapter leadership in Down syndrome research. I think you guys are really doing a fantastic job there.
I hope in the next time when we'll come across this COVID situation, we will arrange one big seminar and you will visit India and Indian chapter of Down syndrome Trisomy Research Society.
That is very tempting. So I say yes.
Thank you.
From now on.
Okay. Thank you. Thank you, Mark.
Thank you very much.
Thank you. Bye.
Bye-bye.
Bye-bye. Okay, participant, please stay online. Now we move on to our last presentation, that scholars presentation. May I request Dr. Papia Ghosh to introduce our scholar for his presentation.
Okay, am I audible? Am I audible?
Yes, yes, you are.
Okay. So the next session is for Upamanyu Pal, who is the senior research scholar working on maternal risk factors of Down syndrome birth. His special interest is to characterize anomalous recombination profile on chromosome 21. He is going to deliver a very short lecture on polymorphism of maternal genes responsible for recombination as risk factor for Down syndrome birth. So it's over to Upamanyu. You share your screen and please start your presentation. Thank you.
Hello. Good evening to all participants and good evening to all participants and delegates. Thank you all for being here. I am Upamanyu Pal of University of Calcutta, and my topic is polymorphisms of maternal genes responsible for recombination as a risk factor for Down syndrome birth. A brief background before I go to my work. So, at molecular level, the most accepted fact that leads to non-destruction of chromosome 21 is error during homologous recombination. These errors can either occur during meiosis 1, which is the most frequent, or during meiosis 2. So what causes these errors? Many studies prove that if there is total absence of recombination, it can cause non-disjunction. Also, placement of chiasma is also a risk factor. How? If a chiasma, that is the point of contact, near the telomere or near the centromere, it can cause non-disjunction. So keeping the above things into consideration, our objective was to look into the genetic aspects of these errors, as this area still remains unexplored. Our objective was fourfold. First, we genotyped our gene for recombination for variants. Second, we studied the amount of recombination across chromosome 21 long arm, followed by studying the chiasma positioning during crossover event. And last, we studied the deleterious effect of the variants using bioinformatics tools. Just in brief, why SNP study is crucial for population-based study. SNPs, or single nucleotide polymorphisms, are the most useful tools in predicting the risk of developing a particular disease and can be used as a diagnostic markers. So ours was a population-based case control study. The exact numbers are given here in the slide. We first categorized our case samples into two groups based on the stage of error they experienced. So we can see almost 85%, that is this blue area, of the mothers had meiosis 1 error, and the rest 15%, that is this yellow area, had meiosis 2 errors. This data, we can say meiosis 1 frequency is much higher. So our candidate gene was MCM9, and after sequencing the entire gene, we discovered 41 SNPs. In this pie graph, the blue area, this blue area shows the SNPs we found in the promoter region. The red area shows the SNPs we found in exon 2. And the green area shows the SNPs we found in exon 3. And the yellow regions shows the intronic variants. Here we divided our variants into three categories, that is risk, protective, and neutral variants. The risk variance increases the odds for non-disjunction, and the protective variance decreases the odds, and the rest are neutral. The red region, this pie graph shows our 25 risk variants.
We cannot hear.
Hello?
Hello?
Hello, can you hear me?
Now we can.
Yeah, you are absolutely audible.
Okay.
Yeah.
Okay. So this red region shows the 25 risk variants. The green region shows our protective variants, and the rest is neutral. From this study, it is clear our sample has a much more higher frequency of risk variants than the protective or neutral variants. And this is the chromatogram showing the 8 novel variants we found in our MCM9 genes, and we submitted them to NCVI and received an ascension number for each. Now, this bar graph shows the comparative frequency of the risk, protective, and neutral alleles in our case groups and control group. We can see the frequency of the risk allele, that is this blue bar right here, is much higher in the M1 case samples than the M2 error group. However, for the protective case samples, we see the frequency is much higher in the control group, that is this green portion, green bar right here. From this data, we can say that our case samples have a significant portion of risk allele that may cause the meiosis 1 error, statistically significant. But we did not find any significant association with the M2 group, and therefore the analysis was done using the M1 control group only, M1 case and control group. Here we performed the genotype analysis in the M1 group for all the 3 variants. In this following bar graph shows the risk genotype for the case and control groups. The case are the red bars and the control are the blue bars. This data shows that the genotype of these SNPs have a higher frequency in our M1 case samples. However, for the protective case samples, we see the frequency is higher in the control group. That is the blue bars and is the control group. Now, here we have shown the amount of recombination in the case mothers with the variants in their genes. This was done by studying markers located in the long arm of chromosome 21 of the M1 case mothers. In these x-axis of the graph, we have shown the amount of recombination events. That is 0 for no recombination and 1 and 2 for the single or double crossover events. Now, we have further divided the case mothers into 3 groups, age groups: young, middle, and old mother, old age group. We see these in this graph that for all age group, the amount of acosmic recombination, that is this red line over here, this amount of acosmic recombination is much higher than the single or double crossover product events. Therefore, we can say this risk variance increases the frequency of non-crossover events, and this happens in an age-independent manner. Now, this bar graph describes the chiasma formation of chromosome 21 QR. The intervals are nothing but the set of markers spanning the entire length of chromosome 21. The interval 1 is a region in centromere, while interval 6 is a region near the telomeric region. When we divided into age group, we found out in younger mother, the telomeric region showed a much maximum recombination activity, and as the age progressed, we noticed a shift towards the centromeric region. However, the risk variant does not appear to significantly alter the chiasma positioning during crossover events. And finally, to explore the damaging effects of the variants, we screened our 25 risk variants with 5 different software. They are Mutation Tester, Polyphene2, SIFT, Provian, and HSF, and their scores for plotting this bar graph is shown here. I have color-coded these variants according to their types. The blue bar shows the promoter variants, the purple ones is the synonymous, the red ones is the missense variants, and the yellow and green ones are intronic variants. Now, from this, it is clear that the missense variants are the most damaging, as they can alter amino acid sequence and disrupt the protein functions. So in summary, we can say the MCM9 gene located in chromosome 6 has a higher frequency of risk SNPs, and this risk SNP can alter the normal splicing process. As a direct result of alternative splicing, the defective protein during recombination does not help in formation of the chiasma between homologous and ultimately fails to segregate properly. That results in direct non-disjunction of chromosome 21. So in conclusion, we can say the risk variants of MCM9 gene are significant risk factor for non-disjunction during meiosis 1, but not meiosis 2. The presence of risk variants significantly increases the amount of acosmic recombination across all age groups, but it does not alter the observable chiasma positioning during recombination events. And in silico analysis shows that the variants affect regular splicing mechanism and can produce alternate splice variants and disrupt gene expression of MCM9. I would like to thank Department of Zoology, University of Calcutta, Department of Science and Technology, West Bengal Government for funding our project, Indian Council of Medical Research for my own fellowships, all the physicians, collaborators, and participating families, and of course, T21 Research Society Netherlands. I'd like to thank my guide, Dr. Sujoy Ghosh, and Dr. Shumantra Sarkar for providing us with the case and control samples, and our 2 overseas collaborators, Professor Stephanie Sherman and Eleanor Feingold, for their constant guidance, and of course, my cytogenetics and genomics research families for their constant love and support. And thank you. And it's open for discussion and questions.
There is a question. I don't know. It is for Upamonu or Mara. Time, it's 7 and 2. Yes, it is for Upamonu. Have you had a chance to look at telomerase activity?
Yes, actually. The telomeric activity is actually found in the younger mother age groups, that experience meiosis 1 error. We can see that in the younger age mothers, the telomeric, most of the chiasma is formed in the telomeric region, and this makes them more susceptible to non-disjunction.
No more question, I think.
Okay.
No more question right now, I think. So, can we conclude the session? Dr. Ghosh can just announce the end of today's webinar, part of today's part. I'd like Dr. Ghosh to please announce the end of today's part of this today's international webinar.
Thank you. Now we are at the end of the first day, and thank you participants for being with us, and keep your patience for the last. And though some of you have already left the meeting, but still we will get back tomorrow, and you please join the webinar a little ahead of our scheduled time. Please enter the Zoom platform at 4:45, and we will start sharp at 5 p.m. Thank you. Thank you once again. Have a nice evening. Thank you.